I am Dr.S.Radha, Assistant Professor in Department of Microbiology of the Standard Fireworks Rajaratnam College of Sivagasi. So, continuing our early discussion on introduction about forensic science, today we will see into the DNA profiling technique. So, this module was designed with the learning objective of what is DNA profiling and what is included under DNA profiling. And next we will see about the methods, how to perform, and finally the application of profiling technique. So, first we have to know as a definition DNA profiling, it is the specific pattern of DNA what we obtain from any samples of bodily tissue obtained from a that is living system. So, it could be blood, semen, saliva, even hair can be used for extracting the DNA and for profiling technique. So, this DNA, if you are considering or comparing with others, most of the DNA part will be similar, and the individual will be having some unique sequence that are known as polymorphic sequence that is taken for study. So, the genes what we are using for DNA study is the having, that is among the 99% of similarity, only few are unique. So, these genes are known as junk genes. If you are taking the human genome, that is it possesses numerous non-coding sequences. So, these non-coding sequences are sequences that doesn't code, that is code for any protein. So, they are inherited from one generation to another generation, and they have many repeated sequence. So, the DNA profile, what is that means? It can be made with any sequences, that is short tandem repeat. For example, these repeats are located around 10 or more sequence or place in the loci, and this repeat have 2 to 5 base pair, that is VNTR repeats. So, these are a length of 9 to 80 base pair. Other than that, there are single nucleotide polymorphism and restriction fragment length polymorphism technique is available. So, SNP, what is that means? It is a single nucleotide variation that is observed among the different population of the individual. So, this is used for studying or comparing the population while considering the family tree. So, here I have shown that the one nucleotide variation among the 4 sequences available. So, this is the diagram that shows the profile, that this profile also shows the variation of male and female. So, we can identify seeing the profile whether they have the X or Y chromosome. So, we are diploid. So, we have all the 2 set and the banding pattern and that is the sex also determined with this profiling. So, here I have shown the steps for DNA fingerprinting. So, you can see first that what we need, we have to isolate the DNA. So, DNA what we are isolating has to be digested with the restriction endonucleases, and these sequences has to be fragmented, that is through electrophoresis technique we have to observe the profile, and this has to be transferred to the nylon membrane. After the hybridization process, the probe what we are adding is vary. So, here I have shown that is VNTR probe is used. So, this probe will be recognized by the technique of autoradiography, where that will be recorded on the X-ray film. So, this is used for studying the forensic inquiries. For example, by comparing the suspects, 2 suspects in the crime scene, that is the scenes whose is matching completely, we can depict the who has done the murderer, and it is also used to solve the disputed parentage problem, that is by comparing the profile of the children with the parents. So, here I have shown the diagram where the suspects are 3 and the crime, that is the scene, they have obtained the bloods from the other than victim, one blood sample is obtained. So, by comparing the pattern what they have obtained, the profile, we can find the murderer, that is a 2. So, again the process I have shown here, that is they have to go for extracting the DNA from the sample, that is the blood or anything, then the restriction digestion, electrophoresis technique, and blotting process by which it is transferred to the hybridization that is membrane, and then that can be by autoradiography, the result will be obtained on X-ray film, and by obtaining or comparing the profile we can find accurately who has done the crime or murder. So, other than these 2 earlier application, an important application is to find the commercial crop or to protect them or to identify the new ones. So this is also used for analyzing the family pedigree. That is, if you are breeding the two new strains or cultivar, using this marker as assisted selection, we can compare. For example, in the diagram I have shown, the marker pattern that is used to compare the susceptible to disease and the new strain that is insusceptible to disease. Then for studying the evolutionary relationship among the two groups of species, for example, the structural and morphological changes of the two individuals can be compared, and the taxonomically new strain, how it is evolving, how it is forming, we can discover and relate them with the earlier one. For example, I have shown here the two squaw type. These are the birds of oceanic environment, that is polar squaw, and another is of brown squaw. So these are available in a polar region. They have seen that by comparing the gene profile, that is PV47-2, that is used as a marker or for DNA profiling. That is both in male and female there are certain X-linked or sex-linked character that can be have this profiling and similarity. So the evolutionary relationship can be studied with this technique. So as a conclusion, I want to tell that DNA fingerprinting is an essential tool for determining and identifying the species sample and assess the relationship between the sample. Thank you for watching this video.